what we're going to do now is we're going to look at the morphology and development of the sponge or the phylum Porifera. So the structure basically and the development of the adult sponge. So we'll talk a little bit about reproduction, but mainly we're just going to talk about the body of the sponge and what it consists of, different types of cells that it has and all of that. So we are, just to remind you, we are in the domain Eukarya. The domain Eukarya includes, remember, the protista, the fungi, the plants, and the animals. So we are now in the kingdom Animalia. And we're going to start with the simplest animals, the phylum Porifera. Now, these animals don't even have tissues, so we can't call them diploblastic because they don't have two tissue layers, because they don't even have one tissue layer. They are not composed of tissues. They do have some specialized cells, but they're not organized into tissues. So let's look at the overall body of the sponge first. And so I'm just going to draw, this is like a cutaway, and then, all right, so I hit my recorder. But anyway, this is like a cutaway, and then let's see. So it's going to come down like this, and kind of this will be kind of a three-dimensional. So what you're looking at is all of this is inside the sponge. All of this here is inside the sponge, and then this is the opening out the top of the sponge. And all I'm going to draw, so the sponge is basically an open cavity. And I'm drawing these, making these pores bigger than what they really are. But water is going to flow in the sponge through the pores, and those pores are called ostia. And then water will flow out the top of the sponge. The opening is called the osculum. Okay, so now we're going to look at just a single piece of the sponge. So let's just say I'll take red and make a little box. We're going to make a little box here. And we're going to open it up. And we are going to draw the different types of cells of the sponge. The way the sponge is organized. Okay, so the cells on the outside of the sponge, which would correspond, I guess you could say, to our skin, are flattened. We're going to use purple to draw them, and I'm going to draw them way over on this side. Pretty much connected to each other. And then some of those form the pores. Okay, and just to make sure that they look like cells, we'll make sure all of these cells have a nucleus. And since I used purple to draw them, I can put their names. They are called pinacocytes. So the cells that line the outside, the exterior of the sponge, the part that comes in contact with the water or with the external environment, they're going to be pinacocytes. Start over. Pinacocytes. Make sure you can see that there. There we go. They're pinacocytes, and then you're going to have like a space in there, and that little space is called the mesohele. You don't have to know that, but there's a little space in there between the pinacocytes and the cells called choanocytes. but that hole that goes through here is your ostium, your ostia. So that's the ostia. I don't know where I would write it because I've kind of boxed myself in here, but that hole is the ostia, just like these pores are the ostia. So I'll do a purple arrow to one of these pores so you know that the pores are the ostia. And they're formed from cells that make up the outside of the sponge, the pinacocytes. And then you have, in the mesohyl, you have amoebocytes. In this little space here called the mesohyl, you have amoebocytes that are not, they don't have like a uniform shape, kind of like an amoeba. And they function in digestion. They're going to take in the food that is filtered by the sponge, and then they're going to break it down and digest it. And in this mesohyl, there are also these silica spicules. Silica is glass, and there are these spicules that are like, they have, it's like a star, but it only has 3 parts. So here's one, and we can draw another one here. Okay, so the orange ones are called spicules. And they help to give the sponge its structure. They're composed of silica, so we call them silica spicules. And silica is really the same component as you find in sand and glass. And then we have our inner layer of cells, and these are the ones that remind us of the protista choanoflagellates. These are the cells that make the sponge so closely related to the choanoflagellates. So we're going to draw them. We're just going to pretend there's like a border here, and they're going to look kind of like this. And then they're going to have flagella. And they're going to form this hole. I didn't mean to close that circle in. Okay, so these in blue are called the choanocytes. And their purpose is, since they have flagella, their purpose is to, let me pull this down so you can see that word. All right, their purpose is to filter food particles from the water that enters through the ostia. So water is going to enter, try to keep my hand out of the way, water is going to enter through the ostia in between those pinacocytes, and then the water is going to flow up, and as it flows up, these flagella that are connected, that are part of the choanocytes, are going to filter food particles, and those food particles will end up inside the amoebocytes. So let me get a black one and write amoebocyte somewhere. And the amoebocytes are going to digest the food. They're going to ingest it by phagocytosis and then digest it. So, I need you to make a drawing of the sponge, and then I need you to answer the questions about the sponge, and you can either look those up, you can Google the questions, or you can use your book, or a combination of both.